Today I will be talking about the role of 3D chromatin organization in gene regulation, and I'm going to walk you through a couple of stories from the lab, including the one that Sanjita mentioned. It would be nice if we keep this informal, so if you have any important questions during the presentation, feel free to interrupt me, and I don't necessarily have to finish the entire presentation. We can talk about the key points. I just want to make sure that everybody can follow. So these are disclosures not related to the talk today. Sanjita also mentioned precision medicine at NYU Langone. This is a new division in the Department of Medicine that I'm co-directing with Dr. Grams. And precision medicine, as we all know, is data-driven. And so the way we think about precision medicine at NYU is through 4 different pillars of data, starting all the way from clinical history. So with electronic health records, we've been accumulating digitized data on lab tests and clinical history on our patients, and then moving on to medical imaging, which includes radiology, MRIs, CAT scan, ultrasound images, but also pathology. So NYU is actually moving to a full digital pathology solution. We've already started digitizing all our slides moving forward. So this enables a lot of research that can be done in computational pathology. Obviously, my lab works a lot in multiomics, so the talk is going to be focusing on genomics today. But the last pillar that we're interested in precision medicine is mobile health. And you can think of your mobile phones as data capturing devices about your lifestyle, and then also all these wearables that people started using in medicine. So we believe this is going to become very big in the future. But going back to what matters, I think, what matters is for us to really try to deep dive and elucidate mechanisms in biology. So how genes are regulated, because this will allow us to eventually cure disease in a systematic way. So it is important to know that we have a long DNA molecule in every cell in our body, and it needs to fit in a tiny nucleus. So the DNA is 2 meters long. It needs to fit in a very tiny space, and it could be doing so in a very random way. So you can just fold a long string anyway, perhaps it doesn't matter. But scientists have discovered over decades that the way this long chromatin inside our nucleus folds has regulatory consequences. It's important for gene regulation, in other words. And actually, people have found that at different levels of chromatin organization inside the nucleus, starting from A and B compartment. So if you're not familiar with these terminologies, all you need to remember is that A compartment includes genes that are activated in a given cell type, where B compartment or heterochromatin is more of silenced chromatin, so genes that are silenced in a specific cell type. And of course, this can vary from cell type to cell type. Different genes are activated, different genes are silenced. And then if you keep zooming in into chromatin, you start seeing these highly interacting, highly organized domains, as we call them. They're called topologically associating domains, TADs for short. So essentially what these are, you can think of them as a functional regulatory unit on chromatin, on DNA. It's a collection of genes and their regulatory elements. So genes are regulated both by promoters and enhancers and perhaps other elements that we haven't accurately discovered or characterized so far. But they seem to be forming these highly interacting domains that connect promoters of genes to their regulatory elements. That's important to remember. And then when we keep zooming in, using the appropriate techniques in the lab, we can actually see and uncover specific interactions between promoters and regulatory elements. So these are the 3 different levels of organization we will be focusing on. Again, A-B compartments, topological domain, and specific loops that connect promoters of genes to regulatory elements. And conveniently, there is a method that was published a few years ago now by the Job Decker group called high-C. which can uncover all these three different levels of chromatin organization in the nucleus. It's really a sequence-based method. You run alignment, the usual algorithms with some tricks, but at the end of the day, you can get information about all these three levels of organization. Most of the talk is going to be focusing on these topologically associating domains. These are almost a megabase long on average. They can be as short as half a megabase or a bit less, and they can go as big as 2 or 2.5 megabases. They are remarkably conserved between human and mouse, which is pretty surprising. The boundaries of those domains of these genomic neighborhoods are enriched by a protein called CTCF. So CTCF is known for its insulating function, because what you have to keep in mind is that these topological domains enable interactions within the locus, but they also prevent somehow, through CTCF in this particular case, interactions from neighboring domains. And this is important because, as you can imagine, a certain gene needs to talk and interact to a certain set of regulatory elements in a given cell type, and it should not be allowed to be regulated by regulatory elements that are outside its domain. And these domains can be cell type specific. They don't necessarily have to be invariant across all cell types. So that's the basic idea. So I'm going to start by introducing you to some work we did a few years ago, led by a postdoc in the lab, Andreas, back in the day, and in collaboration with the I-Fundis lab at NYU. We focused on acute leukemia, and we asked a simple question: whether chromatin organization and to what extent it may be transformed in leukemia compared to healthy controls. I'm going to give you a very quick background on TLL. It's T-lineage acute lymphoblastic leukemia, mainly pediatric cancer with relatively poor survival, especially if the kids relapse early. When that happens, it's unfortunate because the kids have to undergo radical treatments, full body irradiation, and bone marrow transplant, and not always successful, unfortunately. So genetically, it's a relatively simple disease. If you think about genetic drivers, Notch is the main oncogene that drives the disease, and it's mutated in more than 50% of the patients. So how does leukemia happen? So think of the hematopoietic system, starts all the way in the bone marrow where you have the hematopoietic stem cells. These differentiate initially into different types of progenitors. We're talking about TALL, so T-cell leukemia. So we're looking at lymphoid progenitors. The lymphoid progenitors migrate to the thymus, and then normally they undergo different stages of differentiation, eventually exiting the thymus as mature T cells. Now, what happens in leukemia and TLL is there's a block of differentiation because of mutations, because of translocations, fusion proteins, genetic lesions, and epigenetic changes that leave the cells essentially stuck at certain stages of differentiation, immature stages, and they keep proliferating and they create the leukemic blasts. And this can happen, particularly in TLL, either early with early thymic progenitors. So this is called early T-cell progenitor ALL or ETPLL, or leukemia can arise a bit later during differentiation, and then it's called more like a canonical TLL. So study design was very simple. We profiled patient samples using the technique Hi-C to really map chromatin organization in a genome-wide scale. At the same time, we used Hi-C to profile leukemic cell lines, so we can do in vitro testing in the lab. But we also used more typical genomic assays like RNA-seq and also chip sequencing for CTCF. Again, to remind you, CTCF is important because it sits at the boundaries of these topological domains, providing insulation from neighboring domains. What we studied was changes in leukemia, both inside these topological domains and the particular interactions between promoters and enhancers, but also across neighboring TADs. And I will explain why shortly. So the first thing we did was to make sure our data is correct. So we did some quality control. One typical way to do this is by doing a PCA plot or a dimensionality reduction plot. And really, this was also answering a question, whether chromatin organization as a readout can cluster different types of cells. And in this case, it did beautifully so. So what you can see here is 3 groups of samples, 3 coming from healthy T cells, from donors, another cluster corresponding to specimens coming from canonical TLL patients, and another cluster distinct from the previous one corresponding to samples coming from ETP ALL patients. So what we learned here is that you can use 3D chromatin organization without any information about gene expression to cluster your samples in a meaningful way. So then we moved on to additional analysis. One type of analysis was to look at chromatin changes within a topological domain, and this would uncover changes between interactions of promoters to their regulatory elements. It's inside their domain. We asked which topological domains are what we call activated in leukemia compared to healthy controls, and which ones are silenced. And so what you need to observe here is that every dot is a topological domain. So this is a volcano plot that I'm sure you've seen before, but every dot is not a gene in this case. It's a topological domain, which is a collection of genes and regulatory elements. We just marked some of these topological domains by genes that are known to be upregulated in leukemia. And we found that indeed some driver genes or important genes for leukemia reside in topological domains that are activated. They have more interactions in leukemia. And to do this more systematically, we defined 3 classes of topological domains, those that are silenced here in blue, those that have no changes in interactions, and those that are further activated in leukemia. And what we show here on the y-axis is the change in expression. So what this tells you is that essentially, at least on average, when you have an increased number of interactions within a topological domain, the genes that reside in this topological domain will be upregulated, and vice versa. And we can do the same analysis with different types of cells. We compared leukemia samples to both T cells and double positives. And this can give you information on different levels. So if you compare to T cells, if you remember from this differentiation diagram that I showed you before, there's certain stages before T cells become mature. So one of them is double positives. And essentially, leukemic cells get stuck in the double positive stage. So if you compare to the double positive stage, you can get information about why are leukemia cells different from their cell of origin, let's call it like that. When you compare to the next stage of differentiation, you're asking a different question, which is, why don't leukemia cells move on to become mature T cells? Why are they stuck in this proliferating state? And is there any way to encourage them to perhaps mature into T cells? So the second finding was doing the analysis a bit differently. So the first analysis we did was within the topological domain. Next, we asked whether the boundaries of these topological domains are disrupted in leukemia. So this was much more ambitious undertaking. We didn't even expect to necessarily find something here. And in fact, we were surprised to see that there are many topological domains that lose insulation at the boundaries. And perhaps remember what I mentioned in the very beginning. These topological domains are important. Why? Because they limit interactions of gene promoters to their intended regulatory elements. But if you lose insulation going from healthy to leukemia, and now you have an expanded topological domain, which now includes more regulatory elements, perhaps an oncogene residing originally in a smaller topological domain and having access to a small set of regulatory elements, now actually will have access to many more enhancers or regulatory elements and can potentially upregulate its expression. So this is what the scatter plot shows you here. It's a and it's an assessment of inter-TAD interaction changes and intra-TAD changes. And what you see, especially on this inter-TAD interaction changes axis, you see that there's certain topological domains that radically change their insulation. I think there's a question, Sanjita?
So I have a question, and as you said that, to interrupt, I'll forget by the time I go there. So to know that this TAD boundaries are disrupted, you do that analysis by looking at the interaction changes, or how do you do it? Or is it like, how is the analysis is done?
So good question. Let me see if I don't have the slide. So basically the way we do this is by looking at two neighboring TADs at any given time. So imagine you're looking at these two neighboring TADs, and what we're assessing is the changes in this square. You can see my mouse, right?
Yes, I can.
Perfect. So basically we're looking at fold changes between leukemia and healthy here in the inter-TAD square. And this is visualized here on the x-axis. On the x-axis, basically, what you see is the changes in interactions in this square between the topological domains. Now, we found through many analyses that we should also look simultaneously at what changes happen within these original, let's say, small TADs. But the basic metric is what happens between these neighboring TADs. Does it make sense? And we're looking for increase of interaction or decrease of interaction.
Yeah, so you're looking at the changes.
Right, yes. So if there is an increase of interactions between these neighboring domains, that means they're not insulated anymore because interactions are permitted for whatever reason that we can go deeper into. And then the opposite thing can happen. You can have a big TAD that then these changes are lost, and then you have this new boundary coming up. So that will be a boundary gain rather than a boundary loss. So it's looking at the problem from the opposite side. So what we did find is several cases where these topological domain boundaries were lost. And you see that as a dot here. And whenever you see a circle, it means that this loss of boundary was concomitant with a loss of CTCF binding. If you looked at ChIP-seq data, you would see a loss of CTCF binding. And again, this is important because CTCF is known to provide insulation between neighboring TADs. So this provides a further evidence that there's causality here. So something happened, CTCF binding was lost, and then insulation between neighboring domains was also lost. So one of the examples we found was MYC. And I'm showing you a few examples here of the normal T cell samples compared to the leukemic samples here. And then if you look at the fold changes of these contact maps obtained by Hi-C, so the interactions of each leukemic sample compared to the normal, once you will see this, let's say, upregulation of chromatin interactions, which is pretty significant. Now, if we then zoom in at what happens at the very middle point, the center point of this big TAD that now happens in leukemia, what you see is precisely what I described before. If you look at normal T cells, you will see a very sharp, very strong CTCF binding right at the boundary between the two domains. And this is something to expect because if you look at T cells, you have two distinct topological domains. So basically CTCF is binding right here, preventing interactions from one domain to the other. Whereas if you look at the leukemia samples, you will see that this CTCF binding is greatly suppressed. In fact, in some cases, completely gone. So this would explain the loss of insulation. So this was great. So what we asked then was whether we can reverse these chromatin landscape changes in leukemia by using inhibitors, small molecule inhibitors. So one typical inhibitor that's used in vitro, unfortunately not in the clinic because it's very toxic, but for studies in vitro, we use Notch inhibition. Remember, Notch is the proto-oncogene in this disease. And basically what we asked is whether we performed high-C before and after treatment in a leukemic cell line. And we asked for different classes of enhancers, what are the chromatin topology changes? So do we see any changes in the interactions between the enhancer and the target promoter? And you see that if the enhancer is stable after Notch inhibition, which means its activity doesn't change, and activity is measured by chip sequencing for H3K27 acetylation. So for stable enhancers, on average, there's no chromatin change. The same thing for random enhancers. But for enhancers that lose their activity, you start seeing that chromatin interactions are also lost. And if you further focus on those enhancers who lose activity and they're also bound by Notch, and remember, Notch is what we're targeting with a small molecule inhibitor, then you see a further reduction. So what this means is you can target a transcription factor, in this case, Notch, and this can have an effect on chromatin organization, kind of pushing it back to a more normal, healthy state. We validated this using 4C experiments. If you're not familiar with this, it's not that important. But I showed you MYC before, and MYC is very important. It's an oncogene for many, many cancers. We did show that its chromatin landscape changes in leukemia. And it's actually many of the enhancers that control MYC are also bound by Notch. So we believed that by targeting Notch, we would also restore the chromatin landscape of the MYC gene. But this was not the case. So this is, again, a 4C experiment showing basically no change. So wherever you see a peak in a 4C experiment, it means a chromatin interaction with a bait, with a viewpoint, which is, in this particular case, the MYC promoter. So the MYC promoter couldn't care less about Notch inhibition, despite the fact that you have Notch binding everywhere here. And it maintained its set of active promoters, which was a bit disappointing. So we went back to the drawing board and we designed a very simple bioinformatics analysis to find a way, basically, to target MYC. And the idea was very simple. So we classified enhancers into those that are sensitive to Notch inhibition. So those that after treatment, they lose Notch binding, but they also lose enhancer activity marked by K27 acetylation. And then another class of enhancers, which was the resistant class of enhancers, those that despite the fact that you lose Notch binding, enhancer activity remains stable. So what we did next was to ask which chromatin binding factors are enriched in the resistant group. And you will see that many, many factors were not enriched, perhaps some of them surprisingly. Even BRD4 was not specifically enriched in the resistant group. But it became clear that CDK7 was greatly enriched. And the idea was to then use a CDK7 inhibitor published previously and repeat the experiment. So this is what we did. We treated leukemic cells with a CDK7 inhibitor, and we compared the chromatin landscape before and after treatment. And we were happy to see that if we focus on MYC in this case, you see a huge reduction of interactions of the MYC promoter, which sits right here, with several of these enhancer elements downstream. Okay, now I'm going to move on to the in silico prediction of chromatin structure. So why would we want to do this? So perhaps during my presentation, I kind of revealed the reasons why we want to do this, because it's costly every time to do the high-C experiment. Not just the library preparation, but also sequencing has to be very deep to uncover these very specific chromatin interactions between promoters and enhancers. So one obvious motivation here is to reduce the cost of measuring chromatin interactions genome-wide, but also the other side of this is to enable genome studies of 3D chromatin organization in rare cell types. So oftentimes we deal with patient samples, and the number of cells available for us to do genomic experiments is limited. So perhaps you want to do RNA-seq, you want to do ATAC-seq or some chip sequencing, but maybe you don't have enough cells to do high-seq, which actually requires 500,000 to 1 million cells. The other motivation for this work is to enable in silico genetic screening. So the idea being that if you have a good model of chromatin organization, and chromatin organization is very important for gene regulation, perhaps you can start deleting certain regulatory elements or putative regulatory elements instead of doing this using a CRISPR screen in the lab, which is expensive and time consuming and cannot really scale. Perhaps we can do in silico genetic screening instead. So we came up with this state-of-the-art architecture, deep learning architecture to make the prediction. It's a transformer-based architecture, perhaps not important at this point to go into details. What was actually more important is to decide what was going to be the input of this algorithm. So we know what the target is. What we want to predict is the high-C matrix, so essentially an image. But what inputs would we want to do? So with all the considerations I mentioned before, you need to be able to do in silico screening. So that means you need to include sequence. And sequence is important because you have all these chromatin binding factor, transcription factor binding motifs that play a role in binding, gene regulation, and chromatin organization in general. CTCF is important because provides information about insulation, as I mentioned before. But we also needed to include an epigenetic signal because this would provide cell type specificity because the genetic sequence is the same from cell type to cell type. What will allow us to make cell type specific predictions is some epigenetic signal, which shows ATAC-seq. Why? Because it's simple to perform, it's relatively inexpensive, and it requires very few cells. Yet it gives you a very nice overview of the chromatin state via chromatin accessibility. We wanted to also validate quantitatively that this is a good approach, and we're not going to be penalized for using very few inputs in terms of performance. So what you see here is the validation loss of our model as it's been trained through various epochs. And what you see overall is that, as you would expect, the validation loss will go down as the model keeps training and fine-tuning its parameters. And what you see with every different curve is the different combination of inputs we tested. And we're actually very happy to see that the combination that we found to be more practical was actually the one that performed the best. So this is the combination of DNA sequence with ATAC-seq and CTCF. So this is what we call the C-origami model. Everything else performed worse. So after training, we wanted to, of course, evaluate the algorithm eventually genome-wide. But before even going genome-wide, we wanted to take a look at a few snapshots of these predictions in certain loci and convince ourselves that the algorithm was producing images that were close to ground truth. So what you see here is 3 different examples of loci in the genome, different chromosomes. Yes, there's a question.
So I do have a question on the data set because the data is quite noisy for the kind of ATAC-seq data. So do you know like how basically did you carry out the pre-processing of the data so that it will be don't have a lot of noise?
You're talking about the ATAC data, the input data, or the
yeah.
Yes, so it's a very important point, and we actually found that this can have a big impact on the quality of the prediction. So what we ended up doing is, first of all, requiring at least 40 million reads. And then, of course, you can look at measures. You can call the peaks, and you can look at how many reads are under the peak. So you can use all the standard measures. But we do require at least 40 million reads in this case. And that goes not only for training, but also for inference. So once you have a model, if you then have a new cell type, you have an ATAC-seq experiment done. But if the quality is not good, of course, you don't expect the algorithm to perform very well. So your point is very well taken. We have to make sure that both the input data and the training output data is of high quality.
Sure, thank you.
Yes, so yeah, moving on. So 3 different examples. What you see is the ground truth at the top row, and then bottom row has the prediction. So overall, you can see that fine details of the structures are predicted. So different dots here are predicted as above, and different structures, stripes or squares. Diagonal line, of course, it's easy to predict. Now, what you may also notice is that the prediction is actually a smooth, going back to the question, the point made before, high-C experiment itself is noisy, but somehow the algorithm, of course, given several examples during training, is able to figure out that much of the signal that it sees is actually noise. So it kind of smooths it quite a bit. Now, the whole point, so all these predictions were done on the cell line that we trained on, just different chromosomes. Now, the real advantage of building a cell type specific chromatin organization model is to be able to extrapolate on unseen cell lines, new cell types. So this is exactly what we show here. Again, this is just one example. We did this for many cell lines. Basically, what you see here is one cell line on the left-hand side, a different cell line in the middle, and their difference on the right-hand side. And what you see, again, on the right-hand side is the actual ground truth as measured by high-C difference compared to the prediction. And again, you can see that even small details and structures are captured in the prediction. Now, you can do this systematically, not just looking at a few examples, obviously, and we did a lot of validation in the actual paper. I'm showing, I guess, the more difficult one here, which is to predict not just topological domains and their changes in insulation and other metrics, but looking at specific interactions. And Tsirigami is our model compared to a previously published model. You can see there is an improvement even if you stratify these chromatin interactions by their length. You'll see that Tsirigami performs better. Now, having said that, although Tsirigami clearly outperforms these methods, it's still far from perfect. So you see that the stratified correlation is at 0.5, which is good and useful, but it's not perfect. So I think there's a lot of room for improvement here. Now, what are the applications of this method? First of all, since it incorporates sequence, it can make predictions on new sequences. And this happens a lot in cancer. So in cancer, you have genetic lesions manifested as amplifications, deletions, and translocations. So I'm showing an example of a translocation here, which happens relatively frequently in T-cell leukemia between chromosome 7 and chromosome 9. So basically, these mutated cells have a new chromosome, which is a combination of 7 and 9. And if you look at the breakpoint of this translocation, so up here is chromosome DNA coming from chromosome 7, and then below the breakpoint you have chromosome 9. What you see in the actual high-C experiment is that there are interactions across this breakpoint, so which never existed in wild type, by the way. So you have promoters on this side of chromosome 9, what used to be chromosome 9, now interacting with enhancers on what used to be chromosome 7. So a completely new set of regulatory elements controlling now this enhancer, this promoter. And then we asked whether, if we give the sequence to Tsirigami, whether it can predict this high-C map across the breakpoint. And the answer is yes. So it captures all the main structures. What I was personally, I've been looking at these maps for many years now, since the lab is focusing on chromatin organization. And when you're used to looking at these maps, you're used to recognizing different features. One of the very subtle features you see in these maps are these stripes, and these correspond to certain biological events, chromatin loop extrusion events. So there are actually real phenomena happening in the nucleus in terms of chromatin organization. And we were impressed that the algorithm not only was able to find the overall structure and these squares and these triangles, but it was able to clearly identify this stripe, which is actually not that clear in the ground truth experiment. So it does perform some additional smoothing that helps detection of these features. So another application is going to the in silico deletions now, is to actually at the level before attempting to build, a large scale CRISPR screen where you delete multiple elements. Perhaps what you can do before that is you can test it in silico. And that's what we tested. So we tried with just one site first. So we deleted a CTCF site, the one I mentioned before at the MYC locus, the one that was lost in leukemia. So essentially we simulated that in T cells. And we compared the prediction of the algorithm before the deletion and after the deletion. So in this fold change map, you see the changes. And what you see is across the point where we deleted CTCF binding, you now see an increase of interactions. And this makes biological sense because, again, CTCF provides insulation. If you delete it, then insulation is lost, and this allows interactions to happen across the two sides. So this perfectly matched the actual CRISPR deletion experiment that we did in the lab. So this was pretty encouraging. And we thought, now that we've successfully done it once, perhaps we should do it at scale. So we have a pipeline that now scans the genome and in silico deletes every few kilobases. Now, once you do that, I'm going to skip this. Once you do that, you can characterize all the regulatory elements or all positions in the genome by their impact on chromatin organization and as a result on gene regulation. And what we found is if you have this impact score now genome-wide, you can build groups of regulatory elements and characterize them by what else is binding there. So what we found, broadly speaking, is 4 different groups. So one group was expected. So what you see here on the first column is the impact score as predicted by the in silico perturbation pipeline. And what you see here is different chip sequencing experiments for different factors and also ataxic. So what you see in group 1 is binding by CTCF and cohesin factors, which makes sense. This is something you expect to see. You expect elements that are bound by CTCF to be impactful in terms of 3D genome organization. So that was a good validation for the in silico perturbation algorithm. Group 2 was interesting because although it was bound by CTCF and cohesin, it was not accessible. So you can see that here, there's no attack signal. So this can potentially provide clues about unknown biology that we need to study further. And then there's another group where you have very high accessibility actually, but instead of having CTCF and cohesin binding, you have the MAZ protein binding, which is again points to new biology. And actually a few papers came out recently discussing the role of MAZ in chromatin organization. And then interestingly, there's another group of impactful elements where there's no accessibility, there's no cohesin or CTCF, there's no MAZ, but perhaps there are other factors. So what can these other factors be? And they can be cell type specific. So we thought not only we have now a method to do genetic screening and quantify an impact score of a certain regulatory element in the genome or all the regulatory elements in the genome, but perhaps we can use this to identify master regulators of chromatin organization. And so this is what the last application is about. Basically, what we have a way to do now is to make predictions using Tsirigami, perform in silico genetic screen, then get this genome-wide impact score, identify regions of interest that have high impact score, and then proceed with an enrichment analysis for different chromatin binding factors, not just the ones that I mentioned before. And you can do that now in a cell type specific way. So here I'm just showing I'm just showing 3 cell types, so 2 leukemia cell lines and healthy T cells. But you can imagine that if you have access to attack sequencing data and DNA sequence data, you could do that on every cell type, on every data set you have. And what this will give you is really a map of master regulators, so chromatin binding factors, that are enriched in certain elements that are highly impactful for chromatin organization. The high impact coming from the in silico perturbation experiments. So you can get now this heat map where different factors cluster together, and they can be leukemia specific, or they can be T cell specific, or they can be universal. And again, we were happy to see that these universal factors point to CTCF and cohesin. This is something known, and it's a good validation for this workflow and platform. Whereas there are others that seem to be specific for leukemia and others that seem to be specific for healthy T cells. Now, why is this important? Because you can imagine that if you target these master regulators of chromatin organization and gene regulation that are specific to leukemia and not healthy T cells, perhaps this is a viable therapeutic strategy. And just to validate that this worked, at least in the leukemia case, what we did is to rank the putative master regulators, or chromatin binding factors, by how much they're enriched in highly impactful elements. And what we found, kind of connecting it to the previous story, is that CDK7 was actually very high on the list and higher than Notch, which is the proto-oncogene. Yes, there is a question.
So
Yes.
Wenqing, I think you can speak now.
Yeah. Hello. I have a question on the prediction part. So could you help clarify what are the input used for prediction?
Yes. So the inputs are DNA sequence, which you can use. I'm sorry. You can use either let's say the patient-specific or cell line-specific DNA sequence, or if you don't have it, you can use the reference genome. Of course, understanding the limitations. The second input is attack sequencing. So you need to measure attack. You need to perform attack on your patient sample or your cell line. And the last input is chip sequencing for CTCF. Now, after we published our study, we realized that, in fact, we can build a deep learning model that predicts chip sequencing for CTCF using attack sequencing as input. So our current models do not necessarily need CTCF. So basically, it comes down to 2 inputs. It's DNA sequence, which can be patient-specific or not, and attack sequencing. Just these 2 inputs.
Okay. I'm asking because for instance, a lot of cases, the attack seq might not be available. I was just wondering, have you tried for instance, training the model with all 3, but in prediction, only give it the DNA information?
Yes. So we have that. If you look at the training loss, it's much worse, right? If you only train with DNA sequence, you're up here. So you're
No, but train it with all 3, but predict with only DNA.
Yes, we've done ablation studies. It does perform much worse, and there's no real way to rescue this, because think about this: if you don't use attack and you only use sequence, you'll make the same prediction on any cell type. It will just be the same. And yes, of course, because chromatin organization is to some extent conserved from cell type to cell type, you will capture those universal features and elements that are true and present in all cell types. Those you're going to capture. You don't even need to do the experiment. There are certain connections that exist in almost all cell types. So those you're going to capture. But what you're going to miss is the cell type specific features. And all the evaluations we did in the paper was really focusing on what's different from cell type to cell type, making sure we capture the cell type specific. Now, And I will go to Sanjita. There is perhaps a solution to this. There's a middle ground. What you could do, imagine you don't have attack sequencing for your patient or your cell line, but you have attack sequencing or single cell attack from a related cell type, or you don't have from the patient, but you have from a cell line model. What you can do is you can train attack sequencing models from sequence alone, and one of them is chrombpnet. You may be aware of this model. So if you train it on a similar cell type, it's not unreasonable to expect that it will be able to extrapolate on your patient-specific sequence. And then you can use that plus the predicted attack to then predict chromatin. So there is a way to move forward, even in this, very, very challenging scenario.
I see. Another question, you mentioned like the 500 base pair systematic deletion. I was just curious, like, do you also see impact of kind of like smaller deletions? Maybe like, did you ever see cases where maybe like even a single base pair mutation could cause kind of like observable and significant changes in the chromatin?
Yeah, it's a good question. We haven't done it at the base resolution, mainly because we thought it wouldn't work. I think we'll need, at least that was our idea back then, we'd probably need much more deeply sequenced high-C data to train with and more cell lines to train with. So we never really tried it. I guess it doesn't hurt to try. It's just, I wouldn't expect a big differences for two reasons. One, because the model does not have, I believe, does not have the resolution yet to go down to a single base. Number two, even biologically, unless perhaps you hit a certain motif, CTCF motif will be the obvious. So if you mutate the CTCF motif, the very first thing I would check, to be honest, is whether this has an impact on CTCF binding itself. And I mentioned before that you can train a model. We now have models, and there's one published, max attack, it's called, where you can predict CTCF just from attack. So that would be my starting point. If you see differences there, then yes, you can go ahead and try to see if there are differences at the chromatin organization. But there are these two limitations. One is biology itself, because there are redundancies. And oftentimes these CTCF sites come back to back, right? So you mutate one site, but then there's a neighboring site that takes over. So that's something to keep in mind. And the second thing is the capability of the model itself to go down to a base resolution. Now, just since I thought about this now, the redundancy, the nice thing now about in silico perturbations is that you can do combinatorial, right? So you can say, let me look at a region where I have back-to-back CTCF sites, and let me mutate all of them at once and see what happens. Okay, so Sanjita has a question.
Yes, it's very, along the line with what Shao was asking. So she asked about the availability of attack sequencing, and I am asking about the DNA sequencing, because many times, at the clinic, we have the exome sequencing. So is that enough?
Yeah, so unfortunately, whole exome sequencing is not that useful, because here we're trying to look at the role of regulatory elements, and regulatory elements are non-coding for the most part. So it's not going to be helpful in that sense. Again,
so you have to have a whole genome sequencing at some depth, right?
Whole genome sequencing, definitely. Although as a first approximation, if you don't think, it depends on the tumor type. If there are not many, if most of the mutations are coding, then it's a good approximation to use the reference genome. The next best approximation actually would be low pass whole genome sequencing, to at least identify big structural changes. So do you have amplifications? Do you have deletions, even translocations? So these are the changes, the genetic lesions that are expected to have the biggest impact on 3D genome organization. Of course, if you have, high quality, deeply sequenced whole genome, then of course you'll get point mutations, indels, many more events to experiment with.
Got it.
So I think we're out of time. If I had time, I would have talked a little bit about our latest work, which is looking at chromatin hubs as regulators of cell identity, but also going into single cell heterogeneity. So perhaps what I can leave you with is just one final application of Tsirigami, which is based on single cell data. So what we've done in this recent work, which is going to come out in the next couple of months, is we used single cell attack sequencing to profile leukemia patients. And actually, in the beginning, we didn't think that we will see any heterogeneity because most of these patients in leukemia, at least not in solid tumors, but in leukemia, they're driven by a single clone. And if there's a subclone, it's really tiny. But what we ended up finding is that despite the fact that you have one major genetic clone, you have these subpopulations of cells that have a different chromatin accessibility profile. So what makes sense here to do is to apply Tsirigami at the level of these different clusters, of these different populations. And the way you would do it in practical terms would be to do your clustering of the single cell attack data. And then for every cluster, for example, cluster 7, which happens to be very distinct here, generate the pseudo-bulk attack signal, and then feed it into Tsirigami, and Tsirigami will predict now not just cell type specific interactions, but subpopulation specific interactions. And then you can proceed with in silico experiments that are done per clone or per subpopulation. So I'm gonna stop here and thank everybody who contributed to this work, and thank you for your attention and your great questions, and happy to take more questions.
Thank you so much, Tsirigo, for this wonderful talk. So this is open, anybody can ask question, but before, when you are thinking, maybe I can start with that. So Tsirigo, you think for this prediction, if we do not have what we understood, that if we do not have the attack data, we can still use the similar cell type data. Did you train your system? So in the cancer, many times, these are proliferating cell. Can you train, if it is not trained, can you take a non-proliferating cell, say that, neuron or some other which are terminally differentiated cell type, but can you still find their or predict their chromatin accessibility?
Mm-hmm. It's an interesting question. Obviously, the closest it is to your training data, to what you use for training data, the better the prediction. If the master regulators that are expressed in your cell type of interest are very different or quite different from what you trained on, ataxic prediction is not going to work, right? Mm-hmm. Perhaps a way around this would be if you at least, for example, is it reasonable to expect, realistic to expect that you will have gene expression data, perhaps instead of attack sequencing?
Yeah.
Okay, so if you have gene expression data for your cell type of interest, but you don't exactly have attack sequencing, what you may want to do, what you would need to do actually, and I don't know if there are such models out there, you would need a model that takes into account the gene expression profile of your cell type of interest, use that in combination with ataxic data and gene expression data from similar cell types that are close to your cell type, and then really try to extrapolate. But you will definitely need a way to tell the model that in my cell type of interest, there are these chromatin binding factors or master regulators or pioneer factors that are expressed to be able to make the prediction about ataxic. Otherwise, it will just more or less repeat the prediction of the related cell type, but it won't be that useful, I guess.
Got it, got it.
Yeah, so it can be quite challenging. But I think, many people are working on these topics, and I'm sure new models are going to come out, and there's going to be obviously more data for different cell types. And I think eventually it's going to be possible to make predictions even in cell types that you don't have all the data for.
That's wonderful. That's why it's so useful to have the prediction and AI, because, when you are thinking about real disease, you may not have access to the, tissue immediately.
Exactly. And also the other aspect of it is, even if you do, you cannot perform CRISPR experiments on patients, right? You need to have a way to test in the lab before even proceeding to in vitro or in vivo experiments. Great.
Yeah, with that,
Thank you
it's
so much.
one question. Yeah, thank you so much for your time, Dr. Tsirigos. We really appreciate it, and we are looking forward to see how we can use your tools and technologies to, better the human health.
You're welcome.
Yeah, great talk. Thank you both.
Thank you.
Have a
Thank
nice
you.
day. Bye.